Hello, good afternoon to all of my students of this e-learning class, which are present to discuss about excretion in this new session. Today I am here to discuss about excretion from NEB course class 12 Zoology. It's me, Kamal Kesi. Dear students, let's know about the excretion here now. The process of elimination or removal of harmful substances from the body is known as excretion. The organs associated with the removal of harmful substances known as the excretory system. Major excretory system is urinary system because most of the nitrogenous waste products, excess water and other toxic substances are thrown outside as urine. Skin, lungs, liver, and intestine are known as the accessory excretory organs. Nitrogenous waste products are eliminated by urinary system. Volatile substances are eliminated by lungs. Skin removes salt, water, and fat derivatives. Dear students, in our human excretory system, it consists of following organs. They are a pair of kidney, right kidney and left kidney, a pair of ureter, urinary bladder, urethra. Here is a figure of urinary system of human being. You can see that left kidney, right kidney, renal vein, renal artery, urinary bladder, ureter, and urethra too. Now know here about the kidneys, a pair of kidneys. His kidney is dark red bean shaped, measured about 11 centimeter long and 150 gram weight. They lies either side of vertebral column of lumbar region. Right kidney is slightly lower than left kidney. His kidney is enclosed by renal fascia, adipose capsule, and renal capsule. Blood vessels, lymph vessels, nerves, and ureter enter or leave from hilus renalis of kidney. These are structure. You can see this figure also. Microscopic structure of kidney consists of two distinct regions, that is cortex and medulla. Cortex is outer dark part of kidney. It consists of Bowman capsule and nephrons. Medulla is made up of conical medullary pyramids. It is about 15 to 16 in number. Between the pyramids, cortex extend as renal column of Bartini. These medullary pyramids are connected with minor calces, then major calces. The calces open into renal pelvis. Dear students, there is a TS of right kidney. That outer part you can see that a renal fascia. That below the renal fascia there is adipose capsule and renal capsule you can see. Cortex region also you can see easily. That renal column of Bartini there is showing in the figure. Minor calca, that is about 3 to 13 you can see here. Major calca, which is about 2 to 3. Hilum, renal artery and renal vein, renal pelvis, ureter, and renal papilla. Please, dear student, draw this structure well and practice for exam 2. Ureters. These are one pairs. Ureters are thin and muscular tube of about 30 centimeter long. These arises from hilum of kidney and open into urinary bladder. Upper portion is renal pelvis and lower is ureter proper. Let's know about urinary bladder. Urinary bladder is thin muscular elastic bag located in abdominal cavity. Longitudinal fibers and circular fiber make the bag expanding. The collection of the urine in the bladder and discharging from the urethra is called micturition. Internal sphincter and external sphincter control the micturition. Internally, the bladder has a triangular area called trigon in which 3 openings are open. Two are open of ureters and one is opening of urethra. A urinary bladder can collect about 300 milliliter urine. Now, turn of urethra, it is a short canal. Its length is about 20 centimeter long in male and 4 centimeter in female. It is called the urinogenital duct in male and is served for passage of urine and semen. Now, let's know about the structure of the nephron or the uriniferous tubule. Nephron are the structural and functional unit of kidney. These are about 1 million in each kidney. Nephron divided into two parts. They are Malpighian body and renal tube. There is a structure of nephron, that cup-like structure, bowl-like structure. You can see that is a Bowman capsule, and PCT, proximal convoluted tube, that coil tube there is. Descending limb and ascending limb form U-shaped structure, that is loop of Henle or Henle's loop. And again coil tube, there is distal convoluted tube, which is connected with the collecting duct. Numerous 1 million nephron connect with the collecting duct and collect the urine in the urinary bladder. Malpighian body, it has a cup-like Bowman's capsule and a network of the blood capillaries called glomerulus. Bowman capsule is a small double-walled cup. Outer layer is parietal and inner layer is visceral. Visceral cells form passage of fluid to filtrate into Bowman capsule. Glomerulus is a blood capillaries network in Bowman's capsule, entering as afferent renal arteriole and exits as efferent arteriole. The capillaries have small pores. Renal tubule, it is collecting tubular located behind the Bowman capsule. It consists of 4 parts. They are proximal convoluted tubule, PCT, loop of Henle or the Henle's loop, distal convoluted tubule or DCT, collecting duct or CD. PCT now, internally it is lined with brush bordered cuboidal epithelium. It increases the surface area for absorption. Mitochondria provide the energy for active absorption. It is located in cortex and responsible for reabsorption. Henle's loop, it is middle U-shaped tube. It lies in the medulla. It has thin descending limb and a thick ascending limb. Both limbs are supplied with parallelly capillary system called the vasa recta. These supply nutrients and carry reabsorbed water always. DCT is posterior part and located in cortex. It maintains the concentration of urine. CD, the DCT open into CD or collecting tubule. It is present in the medulla region. The collecting tubule join to form large duct of Bertini and to renal pelvis. These ducts drain all the urine toward the pelvis. About 85% nephrons lies in cortex and they don't have vasa recta. About 15% of total nephrons lies in medulla. These are highly supplied with vasa recta. Now know about the mechanism of excretion or the urine formation. Urine formation complete in 3 steps. They are ultrafiltration or glomerular filtration, selective reabsorption, and tubular secretion. Ultrafiltration, it is the first process of urine formation. It takes place in glomerulus, so called the glomerular filtration. Recharged in 1942 explained the dissolved substances are filtered out into the Bowman's capsule due to the pressure of blood. The afferent arterioles enters the glomerular and exit from the form of efferent arterioles. The useful and harmful substances are filtered in Bowman capsule. These are glucose, amino acid, vitamin, and harmful substances like uric acid, ammonia, creatinine, large amount of salt. These are the low molecular weight substances. Large molecular Molecules like protein, fat, carbohydrate are not filtered in there. The diameter of afferent arteriole is wider than diameter of efferent arterioles, so that more blood enter into the glomerulus and less blood volume exit. Hence, it creates hydrostatic pressure of blood in capillaries and force tends to move fluid out of the glomerulus. It is called ultrafiltration. The hydrostatic pressure of blood in the afferent arterioles is about 70 mm of Hg. About 180 liter of fluid are filtered from plasma, but only about 0.5 liter of urine is produced every day. Selective reabsorption. This is the second step in urine formation. It takes place in PCT and Henle's loop. PCT possesses many broader microvilli for absorption of filtrate. Mitochondria present in it provide the energy for active absorption. When the filtrate flows through the PCT, all useful substances are selectively absorbed from the filtrate to the blood capillaries by the both passive and active transport mechanism. About 50% of the urea in the filtrate diffuse back in the blood capillaries by diffusion and about 80% of water by osmosis. Then much reduced volume of the filtrate passes into Henle's loop. Tubular secretion. It is the thin final step in the urine formation. It takes place in distal convoluted and collecting duct. When tubular flows occur through the distal convoluted tubule, unwanted substances present in the blood such as the uric acid, creatinine, ammonia, potassium ion and hydrogen ion are secreted by the blood into the tubular fluid by the process of active transport. At the same time, sodium chloride and the calcium are moved from the urine into blood to regulate the concentration of the ions in plasma. Water also reabsorbed or secreted in the DCT according to the need of water by the body. The regulation of water is controlled by antidiuretic hormone or vasopressin released from posterior parts of pituitary gland. Here is a figure showing the mechanism of urine formation, that exchange of different chemicals, harmful chemical, unwanted chemical, useful chemical, reabsorption in different parts of the urine formation tube, uriniferous tubule is showing, and urine formation with the different urea, uric acid is also showing here in the collecting duct. Dear student, please try to understand from this figure. Countercurrent mechanism. The two limbs of the Henle's loops are opposite in property and the flow of filtrate is opposite in direction. This process is countercurrent mechanism. The mechanism concentrates the urine due to the water diffusion. Vasa recta are the veins present in between the limbs, act as the countercurrent exchanger. The endothelial lining of vasa recta is freely permeable to ions, urea and water. Role of Henle's loop and role of vasa recta are important for countercurrent mechanism. Descending limb of the loop of Henle's is freely permeable to water but slightly permeable to sodium ion and chloride ion. The wall of ascending limb is largely impermeable to water but fully permeable to sodium ion and chloride ion. As the filtrate passes through the descending limb of Henle loop, water moves out in the interstitial fluid and to the vasa recta. The phenomenon maintains the high concentration of solute in the interstitial fluid. In the ascending limb, sodium ion form the filtrate and chloride ion pass out passively. Concentration of sodium chloride become high in interstitial fluid, deep in medulla and lowest in cortex. The ions drawn out from descending limb and from collecting duct. The ions move out from the ascending limb. The filtrate become progressively hypotonic. Composition of urine. Urine consists of 96% of water and 2% of urea. with 2% of urochromy, urinoid, uric acid, creatinine, potassium ion, and hydrogen ion with phosphates. Urochromy makes the urine yellow color, and urinoid gives bad smell to the urine. Isotonic urine is the concentration of the water in urine is equal to the concentration of the water in blood plasma. Hypotonic urine is the concentration of water in urine is more than concentration of water in blood plasma. Hypertonic urine is the concentration of water in urine is less than concentration of water in blood plasma. Osmoregulation in bony fish. These fish have gills and kidney for the osmoregulation. The fish have hypotonic body fluids. Therefore, there is a constant loss of iron from body. It balances the volume of water and salt by free means. Produce large amount of glomerular filtrate. The large amount of selectively reabsorbed solute passes through the renal tube, so fish excrete hypotonic urine. Freshwater fish can take off sodium ion and chloride ion from water directly to the due to the presence of ionocyte cells in their gills. Osmoregulation in marine bony fish. The fish have hypotonic body fluid. This is there is constant loss of water from body. Marine fish conserve the water and loss ion to overcome dehydration of fish tissue by following ways. It constantly drinks sea water. Kidney lack of the glomeruli, so nitrogenous wastes are secreted directly into renal tubules, and water is absorbed by the osmosis. Hence, isotonic urine is produced in small quantity. Ionocyte cells to expel monovalent anions from body fluid to seawater and divalent cations from feces. Osmoregulation in human. In case of human, hypertonic urine is excreted. This minimizes the water loss from their body. The filtrate fluid is Barmin capsules passes through the tubule of nephrons. Then a large amount of the water and solutes are reabsorbed during this course. During cold month, hypertonic urine is excreted, but in warmer month, hypertonic urine is excreted due to sweating. ADS or the vasopressin is the hormone released by posterior part of pituitary gland. The main function of the ADS is to increase permeability of DCT and CD, due to which reabsorption of the water increases. There are two conditions to balance water in human. When a person takes a small amount of the water or large amount of the salt is ingested in diet or excessive sweating, then solute potential of the body fluids become more negative. The change in the osmotic potential is detected by the osmoreceptor in the hypothalamus and carried to the brain. The brain detects such change in the body, and pituitary gland released ADS in the blood. A large amount of the water is reabsorbed rapidly from the filtrate into the cortex and medulla and passes back into the blood capillaries to maintain osmotic pressure normal, so that urine become highly concentrated and reduced volume of the urine is released from kidney. It is generally called antidiuresis. When a person takes large amount of water or little sweating or extremely lost salt intake in diet, then the solute potential of the blood becomes less negative. This condition is detected by osmoreceptor and carried to the brain. It sends the message to the pituitary gland to inhibit the ADS. In absence of ADS, wall of the DCT and CD are impermeable to water and less water is reabsorbed as the osmotic pressure of filtrate is normal and large volume of diluted urine is excreted. It is generally called the diuresis. The regulation of water by the ADS is an example of homeostatic feedback mechanism. Other function of kidney are regulation of fluid balance, regulation of electrolyte concentration, maintenance of acid-base balance, removal of other substances, kidney secrete renin, kidney also secrete erythropoietin, which stimulates the formation of RBC. Function of skin. It is the barrier agent, the invasion of microorganisms and chemicals. It regulates the dehydration. It barrier the agent UV light, regulate the body temperature. It acts as to regulate the excretion of some substances. It absorbs some substances. It is sensitivity in response to stimuli. Function of liver. Carbohydrate, fat, protein metabolism occur in liver. Deoxification of drugs, production of heat, secretion of bile product, storage of vitamin, breakdown of erythrocytes and defense, inactivation of hormones, synthesis of plasma protein. Homeostasis. The regulation or maintenance of a constant body fluid or internal environment is called homeostasis. The temperature, amount of water and glucose concentration are at almost constant in homeostasis. At the temperature of 37 degrees Celsius, enzymes work perfectly, division of cell and metabolism is also perfect. The term homeostasis was first put forward by French biologist Claude Bernard in 1859. In 1929, American physiologist Walter Cannon first used the term homeostasis and study about it. Homeostasis organs are skin, liver, kidney, lungs, endocrine gland, etc. Skin as the homeostatic organ. Skin helps in temperature regulation. It possesses the pigment cell, sweat gland, and sebaceous gland, which control heat and fluid balance. The melanin pigment helps to balance, to absorb solar heat and increase body temperature. Kidney as a homeostatic organ. Kidney is a chief respiratory and osmoregulatory organ. This will play important role in homeostasis. For example, regulation of water, regulation of salt and ions, maintenance of acid-base balance in body, blood volume is regulated by the kidney. Liver as a homeostatic organ. It regulates the carbohydrate, lipid and amino acid. It regulates the glucose in blood. It maintains optimum temperature. Liver produces the bile and excreted product of RBC. Lungs as a homeostatic organ. It balances the concentration of oxygen and carbon dioxide in blood at the best level for the cell's chemical reaction. Kidney transplantation. Kidney transplantation is the organ transplant of the kidney. The damaged kidney is removed and the donated kidney is placed in the pelvic region of the abdomen and linked to the blood vessels. Dear students, this much only for today about excretion in human, and thank you for listening this whole lecture. Best of luck for your NEB exam.